Dear learners, welcome in the course of Immunology BZYET 141. Basically, it is a elective course for BSc Generals. Dear learner, my name is Dr. Siaram. I am an associate professor, discipline of Life Science, School of Sciences, IGNOU. Dear learner, today I want to discuss very important topic that is known as the component of immune system second. What is the expected learning outcome of today's session? So after finishing of this unit, you will be able to explain the role of B and T cell in adaptive immunity. And you will discuss the role of cytokines and chemokines. After finishing of this session, you will be able to differentiate between humoral and cell mediated immunity. A list of step of the antibody activation. How antibody activation take place, we will discuss in detail. And after completion of this session, you will be able to list the feature of the primary and secondary immune response. And you will be also explain the role of NK cells work as a bridge between the innate and adaptive immune systems. So it is a expected learning outcome today. Dear learner, my session is divided into two parts, part first and part second. Part first, we will discuss the adaptive immunity, various cells which are involved in the adaptive immunity such as B cell and T cell and their activations. We will also discuss the cytokines. They can regulate the adaptive immunity. But in case of the part second, we will discuss in detail humoral immunity as well as the cell mediated immunity, primary immune response, secondary immune response, and we will also discuss how NK cell they work as a bridge between the innate immunity as well as adaptive immunity. So I want to discuss this session part first. The first part first, the adaptive immunity. As you know, the antigen basically it is a virus, bacteria and fungi are selectively recognized by the adaptive immune systems, which when the innate immune system fail to defend. When our innate immune system is failed, then adaptive immunity will work. An innate immune system passes an stimulatory signal to activate the adaptive immunity. Basically, innate immunity, they are able to pass the signals, then your adaptive immunity will be activated. Otherwise, your adaptive immunity will be not activated. Adaptive immunity immune response, adaptive immune response take much time, day even week than innate immune response. It is a more pathogen specific and also has memory cell. So your adaptive immunity is a very specific, very pathogen specific, or they have the memory also against the particular pathogen. Adaptive immune response develop after vaccination or exposure to the pathogenic antigens. Means adaptive immunity, it will take place, it will give response after vaccination or a second time exposure against the pathogens. And it is a difference between the innate and adaptive immunity. You can see in this tables, here is the innate immunity, it is a non-specific, it is a adaptive immunity, it is a specific. So you can say it is a first line of defense, it is a second line of defense, and timeline is 0 to 96 hour. It means 5 to 7 week the it will take. In long term, it is less than 5 to 7 days. It is more than 5 to 7 days. So you can understand here is the adaptive immunity and innate immunity. Here is cell which are involved in the innate immunity, NK cell, microphage, neutrophils, dendritic cells, mast cell, basophils, eosinophils. So these are the cells which are involved in the innate immunity. But in case of adaptive immunity, there are the two type of the cell which are involved. One is the T cell and B cell, or you can say the T lymphocyte and B lymphocyte. What is the antigen dependency? It is a antigen independent. Any antigen come, the innate immunity will provoke. Otherwise, it is totally antigen dependent. If antigen like virus, bacteria, fungi will infect our body, then adaptive response will take place. Here is the example of the adaptive and innate immunity. In case of innate immunity, skin, hair, coat, mucus, membrane, phagocytes, granulocytes. So these are the example of the innate immunity. Here is the adaptive immunity: pus, swelling, redness, pain. So these are the example of the adaptive immunity. So you can just differentiate between the adaptive immunity as well as the innate immunity. What is the characteristic of the adaptive immunity? So I want to discuss one by one the various characteristic of the adaptive immunity. One is the antigenic specificity. Another is diversity, immunological memory, and distinguish between self and non-self. These are the characteristic of the adaptive immune response, adaptive immunity, you can say. The one is the antigenic specificity. Antibodies can recognize even single amino acid differences among the protein molecule. Even if you compare with two protein molecule, one single molecule, single amino acid differences are there, your adaptive immunity, they can recognize it. So you can say it is a antigen specific. Diversity, it allow to identification of billion of the molecules on the foreign antigens. Means billions of billion molecules they can identify with your adaptive immunity. And third one is the immunological memory, that they have the immunological memory. Adaptive immune response remember the antigen that previously activated it and produce a stronger immune response against the same antigen the next time, and distinguish between self and non-self. So your adaptive immunity, they are able to distinguish between self and non-self. Which one is the self cell or which one is the foreign cell, they are able to recognize it. The adaptive immune response protecting like liver cells, it is known as the self antigen. And if the antigen are external or foreign, such as bacteria, virus, that is called the non-self antigen. So these are the characteristic of your adaptive immunity, or you can say the adaptive immune response are there. So there are the four characteristic of your adaptive immunity. Adaptive immunity types. So you can classify your adaptive immunity. It is a two type. One is the humoral immune response, mainly involved into the B cell and T cell, and another is the cell mediated immune response, mainly involved in the T cells. So you can say you can classify your immunity, adaptive immunity. It is divided into two parts. One is the humoral immunity, another is the cell mediated immunity. So in this figure you can see it. Here is the various things which are involved in the various cell which are involved in the innate immunity. Here is the adaptive immunity. In case of adaptive immunity, only T cell and B cell which are involved, and cytokines. Cytokine basically it is a chemical. They can regulate the B cell and T cell. And you can see it here is the epithelial layer and dendritic cells and natural killer cell NK you can say, and macrophages and neutrophils. So these are the cells which are involved in the humoral immune response. So I want to discuss before discuss of the humoral and cell mediated immune response. It need to discuss the B lymphocyte and T lymphocyte. According to name B, in bird the brusa fibrosus is and the organ where the B lymphocyte mature. B lymphocyte maturation occur in bone marrow of the mammal such as the mice and animal. So in case of mammal and mice and human, the B lymphocyte mature in the bone marrow. B lymphocyte display the membrane bound antibodies molecules. It is the different from others, and membrane bound antibody molecules have the same antigen specificity and antigen binding site. So you can say it is a lymphocyte. In this figure, B lymphocyte. In this figure, you can see it in case of the bird. They have the brusa of the fibrosus. Here is it can indicate into the red. Indicate into the red color. It is a brusa fabricus. The B cell is mature in this particular part of the birds. The molecule expressed on the B cell, basically the B cell and their surface, they have the different molecules are there. So here is various molecules which are expressed on the surface of the B cell, and B222 it is a molecule which are expressed on the B cell, frequently used as a marker for the precursor B cell and mature B cells. And CR1 and CR2, it is another molecule which are expressed on the B cells. They are receptor for the complement product to bind. And CD32, it is another molecule which are expressed on the B cells and receptor for IgG. And MSC class second molecules, it is another very important molecule which are expressed on the surface of the B cell. They allow B cell to act as a antigen presenting cells. And CD40 interact with the CD40 on the T helper cell surface. So these are the various molecules which are expressed on the surface of the B cells. I want to discuss the B cell activation, how B cell activate. It is a very important. Without activation, immune response will not take place. So I want to discuss one by one the interaction of the naive B cells. Interaction of the naive B cell antibody with antigen as well as macrophages and T cell cause the rapid proliferation of B cell, leading to the produce of the B cell clone. So you can see it here is in this figure. Here is lymphoid progenitor cells are there. Lymphoid progenitors, they can produce the T cell, B cell, and NK cells. So here is B cell. It will proliferate the plasma cell as well as the memory cells. The B cell differentiate into the plasma cell and memory cells, and plasma cells can rapidly, here is the plasma cell, you can see into the yellow color, and they rapidly secrete the antibodies while the memory cell act as a subsequent exposure to the antigens. And memory cell, after subsequent exposure with the antigen, then memory cell activate, ultimately immune response will take place. So these are the B cell activation. The after B cell activation, the immune response will take place, or we can kill the pathogens, or our immune system can kill the pathogens. Another is the T lymphocyte. It is another lymphocytes which are present in our body. Their role is very important in immune systems, basically adaptive immune systems. Here is T lymphocyte. T lymphocyte mature in the thymus. Basically, our T lymphocyte mature in the thymus. It is a very important organs are there, and they have TCR, T cell receptors. That are membrane bound receptor. So you can understand here is the T cell are there. T cells, they have the receptors. Various receptors are there which are available on the T cells. They are able to the um, recognize the MSC molecule. They present the antigenic peptides. And most T cell can recognize antigen only when they coupled with MSC molecule found on the antigen presenting cell. So these are the antigen presenting cells are there. Antigen presenting cells, basically it is a macrophage, dendritic cell and tumor cells. And several molecules are displayed on the membrane of the T cell. T cell receptor CD45 and CD28. So these are the various receptors are there. The T cell receptor and CD45 and CD28, they are able to express on the surface of the APCs. APCs is nothing, it is a microphage, dendritic cell and NK cells are there. The T cell can be distinguished based on the express of the CD4 and CD8 molecules. The T cell can distinguish based on the express. The T cell express the CD4 and CD8 molecule. It is a very important molecule which are present on the T cell. The CD4 plus, they are recognized antigen bound to MSC second molecule and usually function as the helper T cells. And CD8 cells, they recognize antigen bound to the MSC class first molecule and function as the cytotoxic cells. So these are the very important cells are there, CD4 cells and CD8 cells, and they are recognize the antigen bound to MSC class first and class second molecules. Another thing I want to discuss the activity of the T helper cell and activity of the cytotoxic T cell, which are very important cells are which are, that are found in the in our systems, our immune systems. The class second antigen MSC antigen complex on APC recognized by T helper cell. T helper cell, they can recognize the class second antigen complex. The T helper cell get activated, proliferate to form the clone of the effector cells. And the effector cell produce the cytokines and cytokine stimulate T cell and B cell. So basically they can maintain the cycles or the of your immune response. So basically your MSC class second, which are very important, it is recognized by the antigen presenting cells by T helper cell. T helper cell get activated and proliferate to form the clone of the effector cells. Another is activity of the cytotoxic T cells. So you can see it here, the class first molecule which are involved in this case, the class second is MSC antigen complex recognized by cytotoxic T cells. T cells are activated to form the cytotoxic lymphocyte. Cytotoxic lymphocyte causes release to cytokines and help to eliminate the altered self cells. Another very important topic are there, that is known as the cytokines. I want to discuss. They are able to regulate the adaptive immune response. The cytokines, it is a group of the proteins or glycoproteins. Glycoprotein is nothing, they can attach with the glucose molecule to the protein, a carbohydrate molecule to the protein. Basically it is a low molecular weight and secrete mainly by the white cell. The white cell, they are able to produce the cytokines. Cytokines, basically it is a proteins are glycoprotein, low molecular weight basically. It trigger the signal transduction pathway and lead to alter the gene expression in target cell by binding to the specific receptor located on the target cell. These receptors determine whether or not cell is susceptible to the action of a particular cytokine. The very important receptor which are involved in this case, they can determine the immune response will take place or not. The cytokines, basically it is divided into the different group. One is the their action, it would be different. Cytokines autocrine actions bind to the receptor on the same cell and secrete the cytokines, that is known as the autocrine. Same cell, basically same cell involved to bind to the receptor on the same cells and secrete the cytokines. So it is a action of the autocrine. And another is the paracrine action, bind to the receptor nearby the cells. Uh, it may be another cells and nearby the cells, that is known as the paracrine action of cytokines. And third one is the endocrine action of cytokines. They bind to the receptor located in the distant part of the body from one location to other location. It may be distant body target location, that is known as the endocrine action of the cytokines. The cytokines are involved in the activation of proliferation of many cells, regulate of antibodies and cytokine secretion. After binding of the cytokine to the target cell, lead to increase the expression of cytokine receptor. A lot of cytokine receptor will be generated after binding of the cytokine to the particular receptor. They are able to regulate it, or ultimately immune response will take place. Even a small amount of cytokine secreted by lymphocyte can affect the activity of the many immune cells. Example, cytokine released by T helper cell activate a variety of interacting cell such as B cell, NK cell, macrophage, TC, cytotoxic T cells, and hematopoietic stem cells. So they are able to regulate cytokines, they are able to regulate the different cells, and ultimately they are able to generate the immune response against the particular pathogens. And another thing I want to discuss in this slide, the different feature of the cytokine. What is the feature of the cytokines are there? The cytokines, it is a pleiotropy. The some cytokines has different biological effect on the different target cells, like IL-4 secreted by activated T helper cell goes on the activated B cell, thymocytes and mast cell. So you can say it is a pleiotropy of the cytokines. Another is redundancy. It is another feature of the cytokine, that is known as the redundancy. Two or more cytokines perform the same function, means two cytokines perform the same function. A single cytokine function is complex, like interleukin 2, interleukin 4 and interleukin 5 secreted by activated T helper cell cause the B cell proliferation. So ultimately binding of the cytokines like IL-2, IL-4 and IL, the action is same. Their action, their function is the same. They are able to proliferate the B cells. So, you can say it is a redundancy feature of the cytokines. And third feature is synergy. The two cytokines function is a more significant than the single cytokines. So you can say the two cytokines, they are able to bind the T helper cell, or they are able to proliferate the B cells. So two cytokine function is more significant than single cytokine, that is known as the synergy. It is another characteristic of the cytokines. Antagonism, the action of the one cytokine inhibit the function of the another. And cascade inductions, a cascade occur when the cell produce cytokines and activate the other cells. So it is a different characteristic of the cytokines. And another thing I want to discuss, the nomenclature of the cytokines. Cytokines are principally named based on the cell that produce them. One is the lymphokine, cytokine produced by the lymphocyte. So you can say it is a lymphokines, monokine, cytokine produced by macrophage and monocytes, that is known as the monokines. Interleukin, cytokine secreted by leukocyte, they act on the other leukocyte, that is known as the interleukin. And some cytokines are also called their common name, for example of the tumor necrosis factors. In the function of the some cytokines like TNF alpha, this synthesized by macrophage and have several functions like inflammation, formation of the acute phase protein in the liver, neutrophil activation, and uh, another function is interferon alpha. This secreted by macrophage, it create an interviral state of the nucleated cell, activated NK cell and increase MSC first expression. Interferon gamma and secreted by CD8 cells and NK cell, it serves to activate macrophage and stimulate better antigen presentation and increase the overall expression of the. So these are the function of the cytokines. And interleukin 2, they secreted by T cells and cause B cell and B cell proliferation and activation of the NK cells. So these are the various action of the. Another things I want to discuss, that is known as the chemokines. The chemokines are cytokines, they act as a chemoattractant. The important role of chemokines is the migration of the cell between blood and tissues through the venules. Venules is the small veins are there. So they will migrate from one cell to the other cell through the blood. Chemokines are altered to the development of lymphoid organs and T cell differentiation, and they mediate tumor cells metastasis and function as a neuromodulator into the nervous system. Chemokine receptors, various chemokine receptors are there, that is known as the G protein coupled receptor, are required for the action of the chemokine on the target cells. The mechanism of action of the chemokines: the chemokine first bind to the receptors and initiate of the calcium signaling cascade, and then the activation of the small GTPase, and this lead to the actin polymerization, integrin activation and formation of cellular projection and cell movement are there. So it is a mechanism of the action of the chemokines. Dear learner, I want to summarize part first. Basically, you have learned the adaptive immunity occur after exposure to an antigen either from the pathogen or vaccination. You have also learned the characteristic of adaptive immunity and diversity, specificity, memory cells, and self and non-self differentiations. And B cell also are formed into the bone marrow and to the activated form of B cell and memory cell and plasma cell. T cell also recognize T cell that recognize antigen MSC second molecule functions of T helper cell, and while the antigen MSC first molecule function as cytotoxic T cell. And cytokines are low molecular weight protein and glycoprotein that have regulatory function in our immune system. Dear learner, I want to take a short break. Don't go away. We will back within 2 to 3 minutes.
the world's population has reached 8 billion on 15th November 2022. And we are still counting. I don't know where do we go from here. The predictions are scary. Any idea how many people can Mother Earth sustain? India's growing population is a serious concern, and we are soon going to become the world's most populous country. Wow, number one. Population is, after all, a human issue. The UN proposed Sustainable Development Goals cannot be achieved unless and until we consider the population dynamics.
Dear learners, let's start part second. In this part, I want to discuss the cell mediated immunity, humoral immunity, primary immune response, secondary immune response, and we will discuss how NK cell they are able to maintain the innate immunity as well as adaptive immunity. So, I want to discuss the humoral immunity. Humoral immunity, it depends on the molecule found in the humor body fluids. So, it is you can say it is a humoral immunity, means body fluid. Uh, so, this is evolved through the body fluid. The humoral immunity is mediated by antibodies, complement protein and antimicrobial peptides known as the antibody mediated immunity. So, you can say it is a antibody mediated immunity. It also involves the production of antibodies and cytokines along with the activation of helper T cells. Humoral immunity also involved to the various effector functions. The first one is the neutralization of the pathogens and toxins, classical pathway of complement activations, and opsonization of pathogen and help in the phagocytosis. The phagocytosis will take place in case of the humoral immunity. It will activate it, or ultimately antibody they are able to increase the phagocytosis. I want to discuss the antibodies. It is a immunoglobulins found in the blood, tissues, fluid, and the various secretion of the body. And antibody are globular protein that are synthesized and produced by the plasma cell and formed by the activated B cell. The activated B cell, they are able to release the antibodies. So basically plasma cell are there. The 5 classes of antibodies are found in the body: IgG, IgM, IgA, IgE, and IgD. And the antibodies can bind to the specific target to make immune complex that can clear from the bodies. Antibody also promote the phagocytosis by the macrophages. So some antibodies they promote the phagocytosis by the macrophages. The various steps which are involved in the humoral immunity. So these are the various steps. First step is the B cell production and B cell activations. So you can see it in the B cell production. It occur in the bone marrow. B cell production occur in the bone marrow, and B cell receptors display on their surface antigen specific. So they are able to maintain the antigen specific B cells are there. Mature B cell encountered pathogen in lymphoid organ where they have migrated. So you can see it here is the production of the B cell. In B cell activation, mature B cell encountered antigen in the lymphoid organ. So lymphoid organ, various lymphoid organ which are present in our body, they encountered the antigens. The antigen bind to the BCR, B cell receptor. So your antigen basically they bound to the B cell receptor, which then internalize by the endocytosis. Endocytosis, in my previous lecture I have already discussed it. What is the endocytes? They are able to internalize your antigens through the BCR. The antigen fragments are then presented which the MSC second molecules. The proliferation of the B cells, the T helper cell get activated on the binding of the B cell. The T helper cell they get activated on binding of the B cells. Activated T helper cell release the various cytokine. It is another very important process are there. Your T helper cell, activated T helper cell release the cytokines that go on further to activate B cell to proliferate rapidly. The clone of B cell is formed and these cell form the memory cell or plasma cell. Plasma cells secrete large amount of antibody while the memory cell are temporarily resting state. It get activated on subsequent encounter with the pathogens. So it is a proliferation of the B cell. Several steps which are involved in the proliferation of the B cells. And here is fourth process are there, antigen antibody reactions. The macrophage engulf the pathogens. The antigenic peptide are presented with the MSC class second molecules. Activated T helper cell formed due to the macrophage T helper cell interactions. Another step are there in antigen antibody reaction. Plasma cell rapidly secrete antibodies. Activated B cell form the clone of memory cell and plasma cell. Activated T helper cell bind to the B cell and activate it, and memory cell encountered G0 phase and become activated with the same pathogen. So it is a antigen antibody interaction reaction steps are there. So your antigen antibody they react with the particular pathogen or ultimately they are able to produce the antibodies. Another is cell mediated immune response. The cell mediated immune response is a method of our body that fight against the pathogen such as bacteria and viruses. The foreign antigen are recognized by the macrophage and dendritic cells of the innate immune system. They internalize the pathogen, degrade it, present their antigenic peptides on their surface with the MSC second molecule. So it is a process of the antigens are there. So after processing of antigens, they will display. This MSC antigenic complex is responsible for the activation of the adaptive immune system. And CD4 plus and CD8 plus T cells are formed from the T cell precursor in the thymus. So it is a the cell mediated immune response are there. The MSC second complex on antigen presenting cells. So basically antigen presenting cell they have the complex like MSC class first and class second are there. It interact with the CD4 positive T cells and CD8 positive 8 cell interact. MSC class first found on the tumors or infected cells. And the CD8 T cell transform into cytotoxic cell lymphocyte are responsible for killing of the tumor or infected cell. And CD4 positive cell before the interaction with the MSC class second antigen complex present on APC are called the knife cell. So these are the knife cells are there. They will take a role in the activation of the immune response. And the knife cell on activation can form the memory T cell and other T cell variant, or ultimately they will kill the pathogens. Here is mode of action of the cell mediated immunity. You can see it here is the cell mediated immunity. It is governed by the action of the NK cell and macrophages, and cytokine secretion that stimulate innate and adaptive immunity. The first one is the NK cell. They will regulate or their mode of action through the NK cells, and mode of action through the cytokines, and mode of action through the T cell mediated immunity and T cell, or ultimately they are able to kill the pathogens. Another thing I want to discuss the primary and secondary immune responses. So, what is the primary immune response? Primary humoral immune response is generated when the primary contact with the exogenous antigen. So, your exogenous antigen, it may be virus and bacteria, they get contact with the skins, or your primary humoral response will be generated. It lead to the production of the plasma cells and secrete large amount of antibodies and memory cells. And magnitude of kinetics of primary humoral response is depend on the nature and type of the antigen. So, what is the nature and type of antigen of your pathogens are there? So, it totally depends on there. The magnitude and kinetics of primary humoral response. And the involvement of adjuvant is any adjuvant of your antigen or not. So, it totally depends your primary immune response. It will depends on the adjuvant. And route of antigen administered means various route are there, intramuscular, intraperitoneal, peritoneal, and muscles, and these are the various oral, the various route of the antigen administered. They can affect the magnitude and kinetics of the primary immune response. And species of the organism, the various species of the organisms are there. We can take example of the some pathogen like COVID-19. So it is a different species, but influenza, it is the influenza is another species are there. So it is totally depends on the species of the organisms are there, then your immune response will generate, primary immune response will generate. The primary immune response, the antibody production in the different phase. You can see it. The several activities occur during the primary immune response: clonal selection and clonal expansion of the B cells, and subsequent differentiation into plasma cell and memory cells. So you can see it. It is a several step occur during the primary immune response. The antibody production into the different phase. It is divided into four phase. First one is the lag phase, log phase, plateau phase, and decline phase. So you can see through this figure. Here is the antibody titer are there. It is a time. So lag phase, you can see it is lag phase is very long. It will take 1 to 2 weeks. And when the primary contact with the antigen is the established, the IgM appear and engages to the antigenic element, and later IgG involved. Lag phase is variable. Here is example. Here is lag phase is the variable. It may be very short time, or it may be 1 to 2 days are there. So you can take example, pneumococcal polysaccharide persist for several hours, while the diphtheria toxin lasts for 2 to 3 weeks. So you can understand here is the lag phase. It may be variable. In case of COVID-19, the lag phase it around 5 to 6 weeks. The lag phase, here is the log phase are there, rapid increase of the antibody. You can see it. It is a rapid increase of the antibody titer are there when the time is increases. And plateau, an equilibrium state where the antibody production and decay is equal. So you can see it here is the antibody production and decay of the antibody, it would be equal, then you can say it is a plateau phase. And fourth one is the decline phase. The antigenic stimulation is removed and serum antibody level begin to fall. Here is the decline phase. So you can understand here is the different phase of the antibody productions. Another is secondary immune response. And the secondary immune response is generated on subsequent exposure of the antigen in interaction with the memory cell. In secondary immune response, the lag phase is shorter. So here is lag phase is very shorter. Here is lag phase is longer. But in case of secondary immune response, the lag phase is the very shorter. But the magnitude is the greater and persist longer. And there is a secretion of antibodies IgG and IgA other than the IgM. In case of secondary immune response, the IgM production is very less, whereas the IgG and IgA production is very large. And the population of the naive B cell is much lower than to the more antigen specific memory B cells are there. The production of the high affinity and different antibody other than the IgM is due to the process of the affinity maturation and class switching. So it is another very important process are there. They have the higher affinity and different antibody other than the IgM. It's, so it is a secondary immune response. So you can see in this, it is a comparison of the serum of IgG level in the primary and secondary immune response. Here is the serum antibody titer. Here is the 0 days and 28 days. You can see it here is the primary immune response are there. Primary immune response and serum IgM level. So you can see it is the IgM label in the serum, or it is the IgG label are there. So you can say here is the secondary antigen exposure. After it is a primary antigen exposure, you can see it here is the IgM, the concentration is generated very fast, and then IgG will be generated, and after second exposure IgG concentration will be very high, and there are 100 to 1000 fold increase the antibody concentration as compared to the primary response and predominant antibody in the case of IgG along with IgA and IgE. The innate and adaptive immune system work together. The antigen is a non-self recognized, non-self or foreign molecule recognized by the cell of the innate immune system. This formation is passed to the adaptive immune system. The antigen presenting cells recognize phagocytized and digested into the smaller fragment, and these smaller fragment are transferred to the surface of the APC, and this will act as a signal for the other immune cell activations. So these are the innate and adaptive immune system work together. So basically adaptive immune system as well as innate immune system, they will work together, and then they are able to kill the pathogens. And once the antigen is phagocytized and the phagosome is formed, and this phagosome fused with the lysosome to form the phagolysosome, where the antigen is broken down and attached to the class 2nd MSC molecules. The class 2nd MSC molecule antigen peptide complex is moved to the surface and display the antigen peptide. Ultimately different immune response will be generated, and one of the main cells respond to the T helper cell. These release cytokines and cause the further activation of the cells. Here is NK cell bridge between the innate and adaptive immune systems. So here is, you can understand through this figure. Here is the NK T cells are there. it is dendritic cells and the T cells. The NK cells, they work as a bridge between the innate and adaptive immune system. Both activation of the NK T cells by cognate self ligand. The various receptors are there. They will regulate of the T cell as well as dendritic cells, or ultimately they will work or they will kill the pathogens. Here you can understand, here is the intraleukin 12 mediated signaling from dendritic cell induced by the pathogens. So you can see it here is the IL-12, intraleukin 12 mediated signaling from the dendritic cell induced by the pathogen. In case of the second, these are the required for the initial step for protective immunity. These activation event effectively induce interferon gamma production from the NK T cells. So interferon gamma, they will produce it or they will activate your T cells, and when which subsequently activate other cell types such as NK cell and macrophage, and in the innate immune system, as well as CD4 CD8 positive cells in the acquired immunity system, IL-12R and IL-12 receptor T cell type 1 and dendritic cells and macrophages. So ultimately they will work, your NKT cell, they will work, they will maintain, they will make a bridge between the innate and adaptive immune systems. And another thing I want to discuss, the difference between the naturally acquired and artificially acquired immunity. So you can understand, here is the natural acquired immunity and another is the artificial acquired immunity. The active and passive. So you can understand the active. This is acquired when the antigen entered the body naturally. The host fight it can recover naturally. And in case of the passive, it occur when the antibody is passed from the mother to the fetus through the placenta. Antibody is given to the child by the breast milk. So you can say it is a naturally acquired immunity. In case of artificial acquired immunity, in this case, active, the killed or attenuated pathogens is introduced to the body or host, that is known as the vaccination. So you can say it, the vaccination is a artificial acquired immunity. In case of passive, the performed antibodies given to the individual from and the immune animal or persons. Dear learner, I want to summarize part second. So you have learned the various things like humoral immunity is mediated by the molecule like antibodies, various complement protein and antimicrobial peptides. And cell mediated immune response is the method of the body to the fight against bacteria, virus, cancer cell and graft rejection. And primary response is seen on the first encounter with the pathogen. It is usually has a long lag phase, IgM and primary antibody involved. The second response is seen a subsequent exposure to the same antigens. It has the much shorter log phase and it is a greater magnitude. So dear learner, I hope you enjoyed this session. So you have learned the number of the things. What is the cell mediated immunity, what is the humoral immunity, in this case B cell and T cell, it need to activate, then immune response will be generated. If you want to detail study, please go through the immunology BZYCT BZYET 141 course, or you can clarify each and everything. Thank you, learner.